Also same, is it?
Good afternoon, everyone.
Uh, uh,
This is Dr. Pratibha K.S., Head Department of Biotechnology at KLE Societies S, Nijingappa College, Ajaynagar, Bengaluru. Today, one of my student, my name is Vaishnavi S. From second year CBBt. She is presenting her lecture talk on DNA repair. Now, I request Vaishnavi to present her talk on DNA repair. Over to Vaishnavi.
mm-hmm,
Thank you, ma'am. Good afternoon, everyone. I am Vaishnavi from second year
mm-hmm.
CBBt. I would like to thank Dr. Pratibha K.S. ma'am for giving me this opportunity for doing this presentation and guiding me for this. Today, I am here to present a lecture talk on DNA repair.
Okay.
Before jumping into the topic, let me tell you what we all study in this class. Introduction, types of DNA damage, mechanisms involved in DNA damage, photoreactivation,
Okay. But initially,
excision repair,
even in P2's
mismatch
company,
repair, and SOS
I mean
repair.
the
First
team
of
will
all,
do
let us know
RCS
what
notes, right?
is
So like that.
DNA. DNA stands for
Uh,
deoxyribonucleic acid, which is the molecule responsible for the instructions passing on the instructions from an organism which is needed to be developed to live and reproduce. These instructions are found in every cell and they are passed on from generation to
okay.
other. Now let us get to know what is DNA repair, DNA damage. DNA damage is nothing but the injuries uh,
Hmm.
caused to the DNA by various factors such as drugs or by radiations. In further, we will study the damages caused and their effect.
Hmm.
So, uh, DNA damage is nothing but the injuries of the for the DNA which can cause the change in structure of DNA and which may
Hmm.
cause trouble in replication, transcription and cause of mutation
Okay.
which affects the disrupt or breakage in the base pairs and which will affect in their future generation for sequencing. Now let us know what is types of DNA damages that are caused. There are mainly two types of DNA
Okay.
damages,
So if it's a
endogenous
client,
damage and
sorry.
exogenous damage. So in endogenous damage, they are further classified into oxidative damage, hydrolytic damage, mismatch of DNA bases, and alkylation.
Yeah, but
Types
if
of
for
exogenous damage are
process wise speaking,
ultraviolet
RCA is
radiations,
required
ionizing
for all the
radiations,
P1
and
and
chemical
P2's
agents.
city.
Let us see how these agents will cause the damage. First one is oxidative damage. It refers to the oxidation of specific base pairs such as 8-hydroxy-deoxyguanosine, that is 8-OHDG. If we find the oxidation is happening with these bases, then there will be oxidation happening with these
Okay, so
bases. There will be a conclusion that oxidative damage has been taken place. Hence, it acts as the marker for oxidative DNA damage. Now, it will initiate and it will be increased by the chemical carcinogens. This
no,
oxidative
I did
damage
not
will
check
be
that point in the BUPID, but
enhanced by the
usually
chemical
speaking,
carcinogens.
this
And
process
similar
speaking,
type of
P1 and P2
oxidative
to get
damage will also
incidents,
occur
they
in
will have
RNA
RCS, should
with the
have
formation
RCS.
of I8-hydroxyguanosine, uh, which will cause certain neurological damages. Now, second one is hydrolytic DNA damage. So, in this damage, there will be a process taken place, that is deamination. So, deamination means removal of amino group from amino acid and will be converted into the ammonia. Due to
Yes.
this, there will be a damage in kidney. Or if there is a total deletion of base pairs, so this loss of DNA bases is known as AP sites, that is apurinic sites.
Okay. Okay,
So,
this point.
if the damage is left unrepaired and there will be a slowdown or obstruct in the transcription process. Now, let us study what is mismatch damage, mismatch of DNA. So, it is the defect occurring between the two complementary bases
Yes.
which are aligned adjacently to each other. This this will be observed during the replication of DNA. This damage will be bared by RNA, but it will be repaired quickly by the DNA. If there is a failure in repair of these lesions created causes the mutation in the genes. When there is a mismatch in non-complementary purine pyrimidine bases, it is termed as transduction, and if it is formed between the base pairs purine pyrimidine, it is termed as transduction, and in mismatch complementary base such as purine purine or pyrimidine pyrimidine, it is known as transversion. Next is alkylation. Alkylation. This is the common type of alkylation is also known as methylation. Here the methylene group will be replaced by the hydrogen atom. The alkylating agents is seen abundantly in the environment. These agents are produced endogenously. They are formed as byproducts of cellular metabolism, and they cause cuts or tears in the DNA. These lesions may be cytotoxic, mutagenic, and can be neutral to the cell. When there is a cytotoxic lesion, it stops replication, and there will be a break in transcription, or it indicates for the start of apoptosis. When it is mutagenic lesion, it causes mutations or miscodings. Now let us look at into the types of exogenous type of damages, that is UV radiation. So in UV radiations, UV radiation is of it is of chemical agents results in the DNA strand breakage. Chemical agents uh, mainly results in DNA strand breakage. So how this UV radiation is damage in the form of DNA strand breaks. They involve in cut in one or both the strands of the DNA. Double strand breaks are especially dangerous and can be mutagenic. Since they can potentially affect the expression of multiple genes, UV-induced damage can also result in the production of pyrimidine dimers, where the covalent cross-linkage occurs in cytosine and thymine residues. Most common pyrimidine dimers are cyclobutane pyrimidine dimers and pyrimidine photoproducts are the most frequent DNA mutations found in P53 protein in skin cancers. These all proteins uh, help in skin cancers. Pyrimidine dimers can disrupt polymerase and prevent proper replication of DNA. So second one is ionizing radiation. So in this there is a formation of two charged particles or ions. Ionizing radiations is the energy produced from the natural or artificial sources. It is more energy than non-ionizing radiation, enough to cause chemical changes by breaking chemical bonds. So this effect in damages to the living tissues. So these ionizing radiations are of three main types: alpha particles, beta particles, gamma rays, and X-rays. This includes alpha particles includes protons and neutrons. Beta particles includes electrons, where gamma and X-rays are pure energy of photons. So third one is chemical agents. There is in mainly chemical agents are results in DNA strand breaks. So ionizing radiation create the radioactivity decay, or the cosmic rays causes the breakage in the DNA strands. So low level ionizing radiations include irreparable DNA damage. This will lead to replicational transcriptional errors needed to neplasia and may trigger viral infections. so leading to premature or premature aging and cancer. So DNA crosslinks within the DNA, especially interstrand or crosslinks, can lead to DNA strand breaks. If the damage is undergone DNA replication, cross-linked DNA can cause topoisomerase enzymes to stall in the transition state, and when the DNA backbone is in the cleave state, that is, they will be in a breakage in the DNA backbone. Instead of relieving the supercoiling and resealing the backbone, the stalled topoisomerase remains covalently linked to the DNA in the process called abortive catalysis. This leads to the formation of single strand breaks in the case of topoisomerase 1 enzyme or double stranded breaks in the case of topoisomerase enzyme 2. The DNA strand breaks due to the topoisomerase stalling can also occur during the transcription of DNA. In the fact, abortive catalysis and the formation of DNA strand breaks during transcriptional event serve as damage sensor within the cell and help to instigate DNA damage response signaling pathways that initiate the DNA repair processes. Next one, let's now see how DNA repair takes place. Before knowing why should we need DNA repair system, because the body will go genomic instability where the cell may undergo the process of apoptosis or cell death. If not undergoing the cell death, it might lose the process of cell division, and the process of losing the cell division is known as senescence. If this will be the case, then we will have to face lot of disorders. To overcome this, we need a repair system, and if there is a particular type of repair system for particular type of damage. Now, mechanisms involved in DNA repair systems are mismatch of DNA. We have mismatch repair system. To overcome oxidation, hydrolysis and alkylation, we have a base excision repair system. To overcome UV radiation, we have nucleotide excision repair system. To overcome ionizing radiations and chemical agents which causes the DNA double strand breaks, we have two cases, that is non-homologous end joining, that is NHEJ pathway, and homologous recombination HR pathway. So we'll study about photoreactivation. What is photoreactivation? Photoreactivation is a type of DNA repair mechanisms which is present in prokaryotes, archaea, and in many eukaryotes. It recovers the ultraviolet radiation damage caused for the DNA by using visible light. So this is a light dependent process. In this method, cells recover its DNA after UV exposure induced damage. The UV light is lethal to cellular DNA since it induces structural lesions in the DNA by the formation of pyrimidine dimer. So what is pyrimidine dimer? I'll tell in future. Photoreactivation process removes these pyrimidine dimers without altering their nucleotides in the DNA by using energy which is obtained from the visible light. So how was this photoreactivation discovered? It was first discovered DNA repair mechanism in the cell. It was first discovered by Professor Aziz Sansar, for which he was shared the Nobel Prize with Thomas Lindahl and Paul Modrich in the year 2015. But in early 1920s, the scientist named Hermann Muller showed the mutagenic effects of X-rays. But in the year 1940s, Albert Kellner and he showed the healing effect of the visible light against UV induced damage. So he called this process called as photoreactivation. Further, Renato Dalbeco stated that photoreactivation may be enzymatic process which is dependent on light. So this statement was proved by Stan Rupert and named that enzyme as photolyase enzyme. Photoreactivation is seen only in bacterial cell, but other repair systems are seen both in multicellular and bacterial cell. So mechanism of uh, photoreactivation. Photolyase is a flavoprotein and it contains two chromophores which absorb energy from the visible light, and these chromophores acts as catalytic cofactors, and two chromophores which are seen in photolyase enzyme is fully reduced flavin adenine dinucleotide, that is FADH, and foliated group. So the fully reduced FADH is very essential for the catalytic properties of the photolase enzyme. Photolase enzyme can absorb light and get activated. The light activated photolase enzyme splits the thymine dimers formation through the local free radical mechanism by a transient electron transfer method. First, the photolase enzyme recognizes the thymine dimer lesions and it goes and binds to it, and after that it absorbs the visible light at 300 to 500 nanometer range, which is near to the blue light, and it results the light absorption. It absorbs the light, and FADH cofactor get excited or elevated into their higher energy state, and the excited FADH then transfers an electron to the dimer, and is as a result the interaction of charge separated radical pair, that is FADH plus thymine will be separated. This FADH plus thymine is produced. The unstable pair radical pair suddenly splits, and thus the two covalent bonds between the thymine residues breaks. So how UV radiations is induced? The exposure of DNA with UV radiations causes pyramid dimer formation. Among this, different types of pyramid dimers, thymine dimer is the most common one. When UV is induced thymine dimer formation, the thymine residues adjacently situated in the DNA strand are covalently bonded to each other, which causes the structural lesions in the DNA. Thymine dimer is also called as cyclobutane photodimer or CPD because it resembles the cyclobutane nucleus. When UV light hits the DNA, the double strands which is present between the C5 and C6 of the adjacent thymine residues breaks, and the balance of the valency of carbon will be lost, and hence the covalent bonds are formed between these residues. Thymine dimers induced a bulge in the DNA, and as a result, the normal conformational symmetry of the DNA will be lost, and with the lesions formed. Once the bond breaks, the thymine residues will be recovered. The electrons will be returned to the FADH, and the photolase enzyme leaves the DNA, and normal conformational symmetry of the DNA will be regained. This is how photolase enzyme removes the pyrimid dimer from the DNA in the light dependent fashion. And now let us see what is base excision repair. In this DNA repair method, the double helix is damaged or unnatural bases in DNA will be removed by cleaving the N-glycosyl bond without disrupting the phosphodiester bond. So enzymes involved in this base excision repair is DNA glycolase, AP endonuclease, DNA polymerase, and DNA ligase. So let us see one by one how these enzymes will help in base excision repair. The most cited base excision repair mechanism in the cell is the one which removes the uracil residues which are formed in the DNA by the action of the enzyme uracil DNA glycolase or UNG. Uracil in the DNA is created by the spontaneous deamination of cytosine, and since uracil is formed in the DNA, it will destroy the normal Watson-Crick base pairing, and thus in this it should be removed from the DNA, otherwise it will lead to mutations or strand breaks in the DNA. To overcome this problem, a cell has an enzyme called UNG which removes the uracil residue in the DNA. This UNG enzyme scans if there is any uracil residue in the DNA. If it found, it undergoes a glycosyl bond between the uracil and its ribose sugar, and as the result, the uracil residue will be cleaved out from the DNA without disturbing the backbone, DNA backbone, that is phosphodiester backbone. After this removal, the uracil gap is created in the DNA without the pyrimidine base pair. Hence, this gap or region is called episites. This episite will be recognized by another type of enzyme, that is endonuclease enzyme called epiendonuclease. This epiendonuclease cleaves at 5' region on the phosphate backbone of the episite, and the sugar phosphate is cleaved off from the DNA. Then the repair DNA polymerase recognizes this DNA strand and fills it with the correct nucleotide. In prokaryotes, this DNA polymerase 1 is the enzyme which is involved in base excision repair, whereas in eukaryotes, the gap filling is done by the DNA polymerase beta. Then finally, a special type of DNA ligase enzymes called DNA ligase 3, which is sealed to the nick created, and by the addition of new nucleotide by the DNA polymerase enzyme. Thus, this repair mechanism, the original sequence of the DNA will be regained. Now, mismatch repair. DNA mismatch repair recognizes and repairs erroneous insertions, deletions, or misincorporations of the bases which get arises during the DNA replication and recombination, and repair of some forms of this DNA damage. It plays an important role in maintaining genomic stability and cellular homeostasis. For example, MMR increases the accuracy of DNA replication by 20 to 400 folds in E. coli bacteria. The mismatch repair machinery distinguishes the new synthesized strand from the template or parental to begin this repair processes. Two types of MMR mechanisms have been demonstrated. One is expected to be employed by the eukaryotes and the majority of the bacteria. The other is too specific to E. coli or some bacteria. Types of MMR system: prokaryotic MMR system and eukaryotic mismatch repair system. Prokaryotic MMR system, it is seen only in E. coli, which is essentially composed of four identifiable proteins, that is mut-S, mut-L, mut-H, and UVRS. And second one is eukaryotic mismatch repair. MSH play an important role. It is an homologous to the mutase protein. Five highly conserved MSH, MSH2, and MSH6, and mut-SA is composed of MSH2 and MSH6, and mut-SB, which is composed of MSH2 and MSH3. MHL proteins, which are homologous of mut-L proteins of E. coli, MLH proteins, which are homologous of mut-L proteins of E. coli, and there are three complexes in eukaryotic cells, that such as mut-LA, that is mut-LH1, and PMS2 will be composed of mut-LA, and mut-LB is composed of mut-LH1 and PMS1, and mut-LHG. Mut-LG will be composed of mut-LH1 and mut-LH3 3, in addition of mutSHs and mutLHs, uh, eukaryotes also have wide variety of accessory proteins which that are dispensable for the proper functioning of MMR systems, such as proliferating cell nuclear antigen, that is PCNA, and replication protein A and endonuclease 1. These assess the main MMR proteins in recognizing, binding to, or excision the mismatch of the from the duplex of DNA. So how this repair pathway models work? Mismatch recognition will be take place. At the mismatch point, the DNA is sharply kinked about 60 degrees towards a narrow major groove, and MUTES will detect the mismatch of these double-stranded DNA, and by this conserved phenol X glucomotif, and at the end terminus of the bacterial cell, which is initiated by the MMR machinery. And second one is the requirement of related proteins. Mutel interacts with the activated homodimer from the form of mutes and set up a bridge for other protein complexes of MMR. It creates a link between the mismatch recognition by mutes and the proteins responsible for the excision of mismatch, and stimulates the endonuclease activity of the mutate proteins and formation of ternary complexes. Mutes, mutal, and the mismatched DNA form a ternary complexes, and excision and gap fill will be done by excision uh, EXO1, which is activated by the mute S alpha and will be removed by the mismatch base, and then RPA will be displaced the mismatch base after the excision and replacement of mismatch base are finished, and the activity of AEXO1 is inhibited by mute SA alpha and mute L alpha. So photoreactivation, now let us see about SOS repair. Photoreactivation, excision repair, and the post-reactivated combination repair are generally error-free repair mechanisms. However, there is also exists an error-prone, that is error-induced and mutation-induced repair system, that is called as SOS repair. Error in both complementary strand of DNA would be lethal for cell, that will cause damage in the cell. Thus, information will be lost in the DNA. So SOS repair reconstruct the chemical structures of DNA, but the heredity formation will be lost. Hence, SOS repair causes mutation along with the repair of DNA polymerase 5 help in the SOS repair mechanism. It is also known as inducible repair. It involves more than 40 genes which encodes protein responsible for protection and replication of DNA, as well as repair and also mutation. And the SOS response will be found in E. coli and Salmonella typhimurium and Mycobacterium tuberculosis, etc. But it is not found in any eukaryotic cells, and it is not a single discrete mechanism, but includes many responses, such as the ability to repair the thymine dimers. They induced various prophages. They shut off the respiration to delay septum formation during cell division. These responses are regulated coordinately. And SOS response is induced when the DNA is damaged or when replication of DNA stops and single-stranded DNA is accumulated. Agents such as UV radiations, methyl methane sulfonates, as well as chemicals that damages DNA induces SOS response. REC-A and LEX-A gene are major responsible for this SOS response induction. LEX-A encodes the LEX-A protein, which is a repressor. It binds to the SOS box, which is near to the promoter of the SOS gene, and prevents gene expression. Rec-A protein is nucleoprotein and is always high in bacterial cells. It forms a nucleoprotein filaments on single-stranded DNA, and it protects the further damage of the DNA. And Rec-A protein acquire the protease activities and activates self-cleaved and self-cleavage of Lex-A protein from the sauce box, and now the operator and promoter site becomes free and facilitates the gene expression of the sauce box gene. Thank you. I thank Dr. Pratibha ma'am for giving me this opportunity to speak on this topic, DNA repair system. Thank you.
Thank you, Miss Vaishnavi, for your wonderful presentation on DNA repair and about DNA damage. I hope this will help our other students to go through this particular lecture talk. Thank you everyone for watching this video. Stay tuned with us. Stay safe and stay home. Thank you.